Drug release and dissolution.
Drug release. Drug release is the process by which a drug leaves a drug product and is subjected to absorption, distribution, metabolism, and excretion, eventually becoming available for pharmacological action. Drug release is described in several ways, which will be discussed later, which includes immediate release, modified release, delayed release, extended release, controlled release, and pulsatile release. Dissolution refers to the process by which a solid phase, example a tablet or a powder, goes into a solution phase such as water. In essence, when a drug dissolves, solid particles separate and mix molecule by molecule with the liquid and appear to become part of that liquid. Therefore, drug dissolution is the process by which drug molecules are liberated from a solid phase and enter into a solution phase. If particles remain in the solid phase once they are introduced into a solution, a pharmaceutical suspension results. In the vast majority of circumstances, only drugs in solution can be absorbed, distributed, metabolized, excreted, or even exert pharmacologic action. Thus, dissolution is an important process in the pharmaceutical sciences. The effectiveness of a tablet in releasing its drug for systemic absorption depends somewhat on the rate of dissolution of the dosage form and the deaggregation of granules. Ordinarily, of more importance is the dissolution rate of the solid drug. Importance of drug release and dissolution. In the 1962, dissolved drug was known to be necessary for physiologic action, and it was becoming increasingly recognized that capsule and tablet monographs in which the drug substance had a solubility of less than 1% in aqueous medium should include a dissolution requirement. Also, it was shown that in the 70s, it was shown that in vivo bioavailability standard was not required provided that a satisfactory in vitro in vivo correlation could be established. Therefore, the release or the dissolution ensures bioavailability and product performance if satisfactory in vitro in vivo correlation can be established. There are 5 types of dosage forms that can be characterized by release in vitro. These include solid oral dosage forms, rectal dosage forms such as suppositories, pulmonary dosage forms, modified release dosage forms, semi-solid products such as ointments, creams, and transdermal products. Over the last several years, the pharmaceutical industry, pharmaceutical scientists, and the Food and Drug Administration, FDA, have worked together to improve the guidance available for classifying, studying, and documenting post-approval changes to manufacturing process. The first round of this effort resulted in the publication of several SUPAC, which is Scale-Up Post-Approval Change Guidances, including the initial guidance SUPAC-IR for immediate release drug products, followed by SUPAC-MR for modified release drug products, SUPAC-SS for semi-solids, and PACTALS for analytical lab changes. A number of additional SUPAC documents are in development. Parallel with these efforts, the explicit link between physicochemical properties such as drug dissolution and bioavailability were becoming formally recognized as reflected in the Biopharmaceutics Classification System (BCS), introduced in the 1995. The BCS proposed a straightforward classification of drug products based on their solubility and permeability characteristics. According to BCS أو Biopharmaceutics Classification System, Class 1 is considered for drugs that have high permeability and high solubility. Class 2 drugs, the drugs that have high permeability and low solubility. Class 3 drugs, the drugs that have low permeability and high solubility. And class 4 drugs have low permeability and low solubility. In order to understand drug release, students should be familiar with the following terms. Drug substance, an active ingredient that is intended to furnish pharmacologic activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of a disease, or affect the structure of any function of the human body, but does not include intermediates used in the synthesis of these ingredients. So when we say a drug substance, this means the active ingredient. Drug product, a finished dosage form that contains a drug substance, generally but not necessarily in association with one or more of other ingredients. When we say drug product, this is a finished dosage form that includes the pharmaceutical excipients. Immediate release dosage form allows the drug to dissolve in gastrointestinal contents with no intention of delaying or prolonging the dissolution or absorption of the drug. Then when we say immediate release, then we expect the product to be dissolved in the gastrointestinal tract with no delay or intention of delaying the product to be released. Modified release dosage forms. These are dosage forms whose drug release characteristics of time course and or location are chosen to accomplish therapeutic or convenience objectives not offered by conventional dosage form. These include both delayed and extended release drug products. Delayed release products and extended release products. Delayed release of a drug at a time other than immediately following administration. For example, enteric-coated dosage form, it is intended to delay the release of the drug until the dosage form has passed through the stomach. It is a delayed release dosage form. Usually the objective of a delayed or an enteric-coated dosage form to protect the drug from gastric environment of the stomach with an acid-resistant enteric coating, or also to reduce the stomach irritation. Extended release dosage forms, they are formulated to make the drug available over an extended period of after ingestion. Reduction in dosing frequency compared to conventional dosage forms, solution or immediate release. Usually the advantages of preparing extended release dosage form to obtain effective drug concentration for an extended period of time, to improve patient compliance, and to avoid night drug administration. Controlled release dosage forms include extended release and pulsatile release product. Pulsatile release is a release of finite amounts or pulses of drug at distinct time intervals that are programmed into the drug product. In vitro in vivo correlation is a predictive mathematical model describing the relation between an in vitro property of an oral dosage form, usually rate or extent of drug dissolution or release, and a relevant in vivo response, example plasma drug concentration or amount of drug absorbed. This figure shows the difference between extended release or sustained release rate and pulsatile release rate. In case of pulsatile release rate, there is rapid and transient release of drug with a short time period immediately after a predetermined off-release period or a lag time. This is a lag time, for example. While in case of extended or sustained release, in this situation we have a zero order release rate where the release rate is constant over a long period of time. Dissolution
When a tablet is introduced into the GI tract, the drug begins to pass into solution from the intact solid. Unless the tablet is a contiguous polymeric device, the solid matrix also disintegrates into granules, and these deaggregate into fine particles. Disintegration, deaggregation, and dissolution may occur simultaneously with the release of the drug from its delivery form. Frequently, dissolution is the rate-limiting or rate-controlling step in the absorption of drugs with low solubility because it is often the slowest of the various stages involved in the release of drug from its dosage form and passage into systemic circulation. This figure shows the disintegration of tablets or capsules into granules or aggregates, and the deaggregation of the granules into fine particles. It also shows that the dissolution occurs from the tablet or capsule. Also, it occurs from the granules or aggregates, and also from the fine particles. Once the drug is in solution, it will be absorbed into the blood and other fluids and tissues. Frequently, dissolution is the rate-limiting step in the absorption of drugs with low solubility, mainly in the biopharmaceutics classification system for class 2 drugs. The rate at which a solid dissolves in a solvent was proposed in quantitative terms by Noyes and Whitney in 1897 and elaborated subsequently by other workers. The equation can be written as dm by dt equals dCs divided by H multiplied by Cs minus C, or dC by dt equals ds divided by Vh multiplied by Cs minus C, where m is the mass of solute dissolved in time t, dm by dt is the mass rate of dissolution, d is the diffusion coefficient of the solute in solution, S is the surface area of the exposed solid, H is the thickness of the diffusion layer, Cs is the solubility of the solid, which is the concentration of a saturated solution of the compound at the surface of the solid and at temperature of the experiment, and C is the concentration of solute in the bulk solution and at time t. The quantity dC by dt is the dissolution rate, and V is the volume of solution. Actually, the Noyes-Whitney equation is obtained from the fixed second law with the assumption of a steady state, where we have a straight line relationship, which is the dC by dx equals a constant value, where which is the difference in the concentration. Cs is the concentration in the donor side, while the C is the concentration in the receiver side. In dissolution or mass transfer theory, it is assumed that an aqueous diffusion layer or stagnant liquid film of thickness H exists at the surface of a solid undergoing dissolution. This is the stagnant or aqueous diffusion layer. The thickness H represents a stationary layer of solvent in which the solute molecules exist in concentration from Cs to C. Beyond the static diffusion layer at X greater than H, this is X greater than H, mixing occurs in the solution and the drug is found at a uniform concentration C throughout the bulk phase. At the solid surface diffusion layer interface, which is X equal 0, the drug in the solid is in equilibrium with the drug in the diffusion layer. The gradient or change in the concentration with distance across the diffusion layer is constant, as shown by the straight downward sloping line. This is the gradient represented in the Noyes-Whitney equation by the term CS minus C divided by H. The similarity of Noyes-Whitney equation to Fick's law is evident. Therefore, when C is considerably less than the drug's solubility, the system is represented by a sink condition, and the concentration C can be eliminated from the equation. Okay. In order to obtain release from one side, as you notice, this is a release from one side. Okay. In order to obtain a release from one side, tablet is usually embedded in wax by coating the lateral and one of the flat surfaces, flat sides, with molten beeswax, thereby exposing only one of the flat surface of the tablet for drug release. This is the surface that is exposed to drug release. Or by inserting the tablet into a holder and exposing only one of the flat surface of the tablet for drug release. As we mentioned, when the concentration in the aqueous medium is less than 20% of the drug solubility CS, the system is represented by sink condition. If we consider C to be almost zero, then it can be deleted from the equation. Therefore, dm by dt will be equal diffusion coefficient, cross-sectional area, solubility divided by the thickness of the aqueous diffusion layer. Sometimes we write the K as a constant, which is the dissolution rate constant, and it equals diffusion coefficient divided by the thickness of the aqueous diffusion layer. In the derivation of Noyes-Whitney equation, it was assumed that the thickness of the aqueous diffusion layer and the surface area were constant, but this is not the case. The static diffusion layer thickness is altered by the force of agitation at the surface of the dissolving tablet and will be referred to later. The surface area S obviously does not remain constant as a powder, granule, or tablet dissolves, and it is difficult to obtain an accurate measure of surface area as the process continues. In experimental studies of dissolution, the surface may be controlled, as we mentioned earlier, by placing a compressed pellet in a holder that exposes a surface of constant area. Although this ensures better adherence to the Noyes-Whitney equation through and provides valuable information on the drug, it does not simulate the actual dissolution of the material in practice. Example 13.1 in your textbook, Martin. In calculating the diffusion coefficient and dissolution rate constant K, the application of Noyes-Whitney equation is demonstrated in the following two examples. A preparation of drug granules weighing 0.55 grams and having a total surface area of 0.28 meters square is allowed to dissolve in 500 ml of water at 25 Celsius degrees. After the first minute, 0.76 grams has passed into solution. The quantity D divided by H can be referred to as the dissolution rate constant K. If the solubility CS of the drug is 15 milligrams per centimeter cube at 25 Celsius degrees, what is the Ka? I would like also to mention that this example has a couple of shortcomings or limitations, since the amount passed into solution after 1 minute is higher than the weight of the preparation. Also, granules are being used instead of compressed tablet. Just assume that the surface area is constant and that the amount passed into solution after first minute has given to you only to provide you with the dm by dt. According to Noyes-Whitney equation, for under sink condition, dm by dt equals diffusion coefficient multiplied by the surface area multiplied by the solubility divided by the thickness, where d divided by h is called the dissolution rate constant. dm by dt is given in the question since after the first minute 0.76 grams has passed into solution. Therefore, dm by dt equals 12.67 milligrams per second. And by substituting for the surface area and the solubility, the dissolution rate constant can be determined. The diffusion layer thickness in the previous example is estimated to be 5 × 10 to the power minus 3 centimeters. Calculate the diffusion coefficient d. Usually the thickness of the aqueous diffusion layer either being estimated or being determined using an experiment, by conducting an experiment using a rotating disc instead of a stationary one, and the tablet is allowed to rotate at different speeds. Assume that if we determine the thickness of the aqueous diffusion layer and it was found to be 5 × 10 to the power minus 3, what would be the diffusion coefficient? By determining the dissolution rate constant from the previous example, and by knowing that the thickness of the aqueous diffusion layer, the diffusion coefficient can be determined. Drug release kinetics usually fall into two categories: zero order release and first order release. Zero order release, the process of constant release, which is same amount of drug release per unit of time, is achieved from non-disintegrating dosage forms such as topical or transdermal delivery system, implantable depot system, or oral controlled release delivery. This is a zero order equation, where Q is the amount of drug released or dissolved, Q naught is the initial amount in solution, and K naught is the zero order release constant, T is the time. For first order release, there is a change in the release rate with time, or usually there is a decline in the release rate with time, and this is the first order release equation. This figure shows the difference between zero order release and release rate and the first order release. In case of a zero order, the amount released per time is constant, showing a straight line relationship where there is a constant release rate, while in case of a first order release, there is a change in the release rate with time, and usually there is a decline in the release rate with time. Hixson-Crowell Cube-Root Law. If a dosage form's dimensions diminish proportionally in such a manner that the geometric shape of the dosage form stays constant as dissolution is occurring, then dissolution occurs in planes that are parallel to the dosage form surface, and we use the Hixson-Crowell Cube-Root model to understand its behavior. It is thought that the tablet dissolution occurs in this manner. For a drug powder consisting of uniformly sized particles, it is possible to derive an equation that expresses the rate of dissolution based on the cube-root law, or cube-root of the weight of the particles. In other words, the Hixson-Crowell model assumes that the thickness of the aqueous diffusion layer is constant, but the radius of the particle is changing with time. We have spherical particles, assumes that the particles are spherical, and we have isotropic dissolution, which means that the dissolution, the particles dissolve equally from all surfaces. It assumes that the process is diffusion control, and we are operating under sink conditions. The radius of the particle is not, as I mentioned, is not assumed to be constant when the particles are assumed to be a sphere, as shown in this figure. This is spherical particles, and through spherical, through dissolution, the radius is reduced by dr, This is the reduction in the radius, and the infinitesimal volume of this section lost is dV equals 4 pi r square dr. As we mentioned, the radius of the particle is reduced by dr, and the infinitesimal volume of this section loss is dV equals 4 pi r square dr. This equation is valid if we have one particle. If we have n particles, then the infinitesimal volume will be dV equals 4 n pi r square dr. The surface area of the n particles equals 4 n pi r square. Of course, we are assuming that the particles are spherical, and this is the surface area of spherical particles. The infinitesimal mass change, as represented by Noyes-Whitney equation, equals minus dm equals k s cs v dt. Of course, in this case, the sign is negative because we are measuring the undissolved mass. Therefore, there will be a decrease in the undissolved mass. Minus dm, where the mass equals the density multiplied by volume, then minus dm can be written as minus the density multiplied by the change in the volume equals the solution constant multiplied by the surface area multiplied by the solubility multiplied by the change in time dt. If we substitute for dV and S in the previous equation, we obtain the following equation where dV equals 4 n pi r square dr and S equals 4 n pi r square. Divide the equation divided through by 4 n pi r square, we obtain this equation minus density multiplied by dr equals k cs multiplied by dt. Integration of the previous equation with r equals r naught, the original radius at time, equals 0, and r equals r radius at time equals t, we obtain the following of this equation: radius r equals the original radius minus constant k cs multiplied by the time and divided by the density. When we are conducting a dissolution experiment, it is not practical to measure the change in the radius of the particles with time. Therefore, it will be easier if we measure the amount dissolved with time and then determine the weight of the undissolved powder as a function of time. Therefore, the radius is being replaced by the mass. The radius of spherical particles can be replaced by the mass of n particles. The sphere volume equals pi d cubed divided by 6. Therefore, the amount or the mass equals number of particles multiplied by the density multiplied by the volume of a sphere. If we take the cube root of the previous equation, we obtain the Hixson-Crowell model. M naught, the mass, the weight undissolved, original weight undissolved to the power 1/3 minus the weight undissolved to the power 1/3 at certain time equals constant k multiplied by the time, where k is the cube root dissolution rate constant and is shown in the following equation, in this equation. This is an example of how the Hixson-Crowell model is being applied. A specially prepared tolubutamide powder of fairly uniformly sized particles with a diameter of 150 micrometer weighed 75 milligrams. The solution of the drug was determined in 1000 ml of water at 25 Celsius degrees as a function of time. Determine the value of K, the cube root dissolution rate constant at each time interval, and calculate average value of K. These are the data usually that are obtained experimentally, the time and the concentration
dissolved at each time. For example, at time equals 0, okay, the concentration dissolved is 0, and the weight undissolved should be the original weight, which is 0.075 milligrams. At time equals 10 minutes, the concentration dissolved equals 0.0197 milligrams per ml. And since we have 1000 ml, then the weight dissolved equals 0.0197 grams. The remaining weight undissolved is the difference between the original weight and the weight dissolved at 10 minutes, and it equals 0.075 grams minus 0.0197, and it equals 0.0553 grams. Then we determine the difference between the cube root of the original weight minus the cube root at time equals t. Is that the cube root of 0.075 minus the cube root of 0.0553 equals 0.0406. In order to determine the dissolution rate constant, we divide the difference between the cube root divided by the time, the 10 minutes, and we obtain the dissolution rate constant. The calculations for same manner or same procedure is being used in order to determine the difference in the cube root between the original weight and the weight at time t, and it is divided by the time in minutes to obtain the dissolution rate constant. In the end, the average value is being taken for the 5 values, and it equals 0.00424 grams cube to the power 1/3 per minute. In a situation in which the aqueous diffusion layer thickness about a spherical particle is comparable to or larger than the size of spheres, micronized particles less than 50 micrometer in diameter, the change of radius with time becomes r square equals r naught square minus twice diffusion coefficient multiplied by the solubility multiplied by the time and divided by the density. There are several models that can be used to calculate or to measure the dissolution of particles. One of these models is the Higuchi-Hiestand model. Higuchi's model assumes that the thickness of the aqueous diffusion layer is not constant and that it is related to the diameter of the particle. Thickness of the aqueous diffusion layer is assumed to be equal twice the diameter. Based on this assumption, this equation is obtained. The estimated time for complete dissolution is when the radius becomes 0, can be calculated using this equation, where the estimated time equals the density multiplied by the original square radius, original radius square, and divided by twice diffusion coefficient multiplied by the solubility. This is an example of how the estimated time for complete dissolution is being calculated based on Higuchi-Hiestand model, which assumes that the thickness of the aqueous diffusion layer equals twice the diameter. In clinical practice, diazepam injection is often diluted many folds with normal saline injection. An immediate or incipient precipitation of diazepam occurs invariably upon addition of saline followed by complete dissolution within 1 minute upon shaking, with solubility in water equals 3 milligrams per ml and density equals 1 gram per ml, and diffusivity or diffusion coefficient equals 5 × 10⁻⁻6 centimeter square per second. Calculate the time for complete dissolution when the original radius equals 10 micrometer. The time required for
complete dissolution is calculated since the density is known and the original radius is known and the diffusion coefficient is known and the solubility is known. The time required for complete dissolution, if the original radius equals 10 micrometer, is equals 33 seconds. If the original radius is 25 micrometer, then the time required for complete dissolution will be higher and it equals 208 seconds. Factors affecting drug dissolution. There are several physicochemical and physiologic factors that control the dissolution of drug products in humans and need to be considered when designing dissolution tests. They are the composition, mixing patterns, and volume of contents in the GI tract. Example, the presence of a meal in humans raise the pH of the stomach from its normally acidic state to pH 5.5 to 7. This could affect solubility of drugs, which can affect oral bioavailability. Also, the diffusivity of drug can also be affected by the presence of food. The presence of food increases the viscosity of the GI fluids and reduces the diffusion coefficient of the drug. The boundary layer thickness around the dissolving particle will depend on how vigorous local mixing is. In other words, an increase in gastric or intestinal motility may increase the dissolution rate by reducing the thickness of the aqueous diffusion layer. According to Noyes-Whitney equation, the driving force for dissolution is the difference between drug solubility and the concentration of the drug in the GI fluids. As the concentration of the dissolved drug becomes larger, the driving force is reduced. To maximize drug dissolution, the concentration of dissolved drug must be minimized. This happens when the drug is absorbed through the intestinal wall and into the blood. The pH of the diffusion layer has significant effect on the solubility of weak electrolytes drug and its subsequent dissolution rate, since the pH affects the solubility and this will affect the dissolution. Particle size and surface area of the drug have significant influence on the drug dissolution rate. As we mentioned in previous equations, Noyes-Whitney equation, for example, that by increasing the surface area, the dissolution rate increases. Therefore, the particle size should be taken into consideration. Amorphous or non-crystalline forms of drug may have faster dissolution rates than crystalline forms. Usually, amorphous compounds or amorphous drugs, they have higher solubility than crystalline compounds. Therefore, they will have a better dissolution. Temperature. Drugs dissolve at a more rapid rate on heating because most of the drugs solubility increases for most of the drugs as the temperature is increased. Also, the diffusion coefficient increases by increasing its temperature. Therefore, we do expect that raising the temperature will increase the rate of dissolution. Surface active agents will increase the dissolution rates by lowering the interfacial tension, which allows better wetting and penetration by the solvent. The surfactants can be available biologically in the GI tract, and also sometimes they are added into the dissolution medium.